everyone, I'm Anaid, and today I will be explaining the relationship between a codon and an amino acid, and also how to read an mRNA codons chart. Codons are sets of 3 base pairs that code for a specific amino acid. Amino acids are organic compounds containing amine and carboxyl functional groups, along with a side chain specific to each amino acid. The relationship between codons and amino acids is that a codon codes amino acids. For example, if we were talking about trees and seeds, the relationship between a seed and a tree is that the seed carries all the instructions to make a healthy tree, the seed being the codon and the tree being the amino acid. The codon table may be intimidating to read at first, but it's not too hard to understand its organization. Let's walk through an example. Suppose we are interested in the codon GAG, and we want to know which amino acid specifies it. First, we look at the left side of the table. The axis on the left side refers to the first letter of the codon, so we find G along the left axis. This tells us the broad row of the table in which our codon will be found. Next, we look at the top of the table. The upper axis refers to the second letter of the codon, so we find A along the upper axis. This tells us the column of the table in which our codon will be found. Finally, you can use the third axis on the right side of the table corresponding to the intersect box. By finding the third nucleotide of the codon on the axis, you can identify the exact row within the box where your codon is found. For instance, if we look for G on this axis in our example above, we find that AUG encodes the amino acid GLU.
Hi, my name is Samantha, and I'll be talking about what is a start codon and a stop codon, and what happens in the small subunit. The start codon is the first codon of a messenger RNA transcript translated by a ribosome. A codon is a sequence of 3 nucleotides that together form a unit of genetic code in a DNA or RNA molecule, like the beads in a bracelet, and the bracelet will represent the DNA or RNA molecule. In DNA, a start codon initiates the translation of the first amino acid in the polypeptide chain. The translation would be AUG and methionine. Sometimes it codes for GUG or UUG. In the genetic code, a stop codon is a nucleotide triplet within the messenger RNA that signals a termination of translation into proteins. It is the end of the translation, which codes UGA, UAA, UAG. This small subunit is in charge of information flow during protein synthesis. It initially finds a messenger RNA strand, and after combining it with a large subunit, ensures that each codon in the message is paired with the anticodon in the proper transfer RNA. The messenger RNA is thought to enter through a small hole.
Hello, my name is Ana Karen, and today I'll be talking about what happens in the large subunit. Large subunits contain sites where new bonds are made when creating proteins. The bonds form polypeptide chains from amino acids. Think about it like a chocolate factory, where different machines help create the chocolate bars. The factory are the large subunits, and the machines would be the sites. The sites are called 60S in eukaryotic cells and 50S in prokaryotic cells. S stands for Svedberg unit, which is the name of the scientist who helped create it. For example, if the factories were found in 2 different locations, they would have different names, such as Hershey's and Kit Kats, depending on the person who owns the company. The ingredients such as cocoa, sugar, and milk that is used to make the chocolate will be the different amino acids that help form the chocolate, which would be the polypeptide. Now, let's go with my partner, Vanessa.
Hello, my name is Vanessa Torres, and I will be presenting how to make a protein with 5 amino acids. A protein is what the body needs for building structures and maintaining life. When you break down a protein, you will find a lot of amino acids bonded together. This is because amino acids are the building blocks of proteins. Multiple amino acids are linked together by peptide bonds forming a long chain. Some steps of how to build a protein are the following. First, the RNA moves to the ribosome. This type of RNA is called the messenger RNA. It is abbreviated as mRNA, where the M is for messenger. Second, the mRNA attaches itself to the ribosome. Third, the ribosome figures out where to start on the mRNA by finding a special 3-letter begin sequence called a codon. Fourth, the ribosome then moves down the strand of mRNA. Every 3 letters represents another amino acid molecule. The ribosome builds a string of amino acids based on the codes in the mRNA. Finally, when the ribosome sees the stop code, it ends the translation and the protein is complete. To put it in simpler terms, this is how I did it. First, I got the amino acids that I was going to use to make the protein. For example, I used serine, isoleucine, alanine, and cysteine. Then, I found the codons for each amino acid and placed it in the RNA strand. In this case, I used UCG, AUU, GCU, UGU. The last step will be to find the codons in their DNA form to put them in the DNA strand. In this step, you are connecting them to their nucleobases like adenine, guanine, thymine, and cytosine. Finally, you are left with your protein.
Thanks for watching. I hope we were able to teach you guys something new. Bye!